Great, and I think my talk is the perfect one to follow Barbara, actually, because I'm going to pretend I'm in a world where everything she just said works, and every piece of reproducible data is out there. Where are we going next with all of that, and what do we do from there? So I used to be at Stanford. I will have been at Stanford until April Fool's Day next week when I'm moving to UCSF. Actually, I'll talk about that at the end. So I'm even closer across the bay. First, I got to start with all my disclosures because we didn't talk about conflicts of interest. Barbara talked about a lot of things, but not conflicts of interest. These are all my disclosures. Suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You shouldn't believe another word I say over the next 30 minutes. Wouldn't blame you. But I'm most proud of the bottom right here, which are all the companies started by my students. More than half my graduate students start companies, and they do this with the most amazing platform in the world, and that is data. And it's big data, and it's big open data. I'm going to show you how they do it. I'm going to show you how I do it, and I'm just going to show you how you can do all of this as well. You guys obviously know we're in the middle of this data deluge of genomic data. I show here just the one kind of chip that launched my career 15 years ago. One example of a high throughput measurement modality in genomic sciences. This is a standard Affymetrix gene chip. And it still looks amazing to me. I mean, just the packaging of this thing. Obviously, they're over on my side of the bay, over in Santa Clara, right next to Intel. It kind of looks like an Intel chip. And man, with one of these chips, it's the size of your thumbnail. You can see a picture of a thumbnail at the bottom. You can just quantitate, for example, all the RNA in a genome just on a single chip. And while that is amazing, it's even more amazing that the price has come down to a couple $100s for one of these chips. And you know, every time we keep thinking about newer technologies like RNA-seq and all the rest, there's still a new site installing an Affymetrix machine somewhere. I mean, so when I say microarrays here, though, I do mean RNA-seq. I mean all this data that we're talking about. Of course, when you get tired of measuring these one by one, on the right is the 96-well format of these. So you can just see each well here is a little microarray. I find it humorous that in Silicon Valley, we scale IT kinds of things by 1024, but life science scales with 96, right? You've got to go from 96 well to 384 well, and you can see the multiples from there. But I still think the most amazing part of these chips, I don't know, 15 years later after these came out, really, the most amazing part of these chips is not the chip, right? What happened after these chips came out and these technologies, right? In early 2000, 2001, 2002, everyone started writing papers about microarrays. Here's a list of genes that are different. And this cancer and diabetes and that tissue. Everyone had a list of genes. And then the top journals like Science and Nature, PNAS, and now many of the middle-tier journals, they started to say, hold on a second. No more papers with these microarrays. No more. Unless you deposit your data into an international repository so that someone else can double-check the math. And as we heard, it's getting more important to double-check the math as we get closer to the clinic. And maybe someday someone else can do something with these samples. And the precedent for this goes back to GenBank, which is now 32 years old, right? First figure, no one wants to retype A, G, T's, and C's. You got to electronically disseminate. That goes back to the tape era. Forget about internet, right? And so the precedent says that if enough people are using a molecular modality, you got to have a repository for that. Fast forward from 2000 to 2010 till August of 2012 when we hit a threshold. Now, if you look today, 1.7 million microarrays publicly available. 1.7 million of them. You can just see that you can get to them from Array Express or NCBI Geo, both of which were on Barbara's slides there. And this is still doubling every 2 to 3 years. We hit this magic threshold of 1 million microarrays in August of 2012. Well, that's eras ago, right? 2012, we hit 1 million, now we're at 1.7 million. Just see the growth rate. We're about triple, let's say doubling every 3 years or so. Now, it's so easy to get lost in all these millions and billions, right? I totally agree. I mean, it sort of washes over you. But let me just tell you how amazing this is. And some of you have heard me give this talk, you know what I'm going to say next, right? In the middle of this window here, right, is a simple search box, right? You can see the search box there. Any one of you, forget about you, any high school kid that needs to do a science fair project today, she can go to this website, she can type in breast cancer for a science fair project, and find and download nearly 50,000 digital samples of breast cancer about as easily as she could find a song on iTunes. And that's an amazing thing today. It's not perfect. I know John Ioannidis, he's in my building, right? I know half of this data is still missing. I know we don't agree on the standards. I know we're missing annotations. But you get 50,000 of them now, today. Actually, the number that impresses me more is the number above the 50,000, right? 2,000 independent labs have already submitted data on breast cancer to this, whether it's cell lines, biopsies, dog models, viral samples, 2,000 labs. Let me tell you how amazing that number is for a second. Let's say I'm a brand new researcher in breast cancer, okay? I'm a newbie, and I'm trying to get my friends maybe on the internet to help. That's crowdsourcing, you know crowdsourcing. Let's say I'm a brand new researcher in breast cancer, and I'm writing a grant, and you're all on my NIH review committee. And in my grant, I propose to get 2,000 of the best researchers in the world to get their best characterized patients with breast cancer to run the latest technologies in the world and give me all that data for free, okay? Now, if I propose this in a forward direction, you would laugh me out of the room. Why would 2,000 researchers give you their best data for free? Yet, here it is. 2,000 researchers are there giving you their best data for free. But it's kind of backwards. So I like to think of public big data as retroactive crowdsourcing. 2,000 labs are there to help you, and they don't even know it. Actually, 2,000 labs are there to help you, and you don't even know it. And if for some reason 40,000 samples aren't enough, 50,000 samples aren't enough for you, just wait 3 years. It'll be 100,000 samples. We're adding 100 samples just during my talk. Right now, we're doing nothing. Someone somewhere is supplying the data. And whether it's breast cancer, lung cancer, prostate cancer, whatever disease you want, the data is just sitting there. In some ways, this is my answer to the reproducibility problem. I don't believe any one of those 2,000 data sets. I don't have to, but I believe what's in common across them, right? Maybe more than half are seeing the same gene, the same target, the same answer. That's what I trust now, moving forward. So I do reproducibility backwards. I don't care about any one paper. With all respect to Barbara, I don't even care if any one of these was published in Science. I truly don't care. I want to see what's in common across the best scientists in the world. Now, if you have any doubts that a high school kid can do it, here is the one science kid who's done it, right? The high school kid. This is Brittany Wenger. I still have yet to meet her. She's from Sarasota, Florida. She's at Duke now. And at age 17, she downloaded data and built this artificial, what they call an artificial brain. Of course, it's a machine learning neural network for breast cancer. She had immunohistopath data and some RNA-seq data was public. She was able to design it. And the following year, I guess she was bored. She was in the newspapers for doing this for leukemia. And she won the top Google Science Fair competition back, 3 years ago for this. Now, of course, what I'm saying is if a high school kid can do this, we all can figure out how to do this too. In our own academic institutions, our own research institutions, all this data is sitting there. It's not perfect, but it's sitting there waiting for you because there's nobody else out there looking at this data. Everyone who knows how to get funding to create this data, you know what happens when they deposit it? They get another grant to go measure more things. And they leave this, yeah, they might get a Science Cell or Nature paper out of it, and all this data is just sitting there waiting for something magical to happen. It's up to you to provide the magic. So I'm just going to show you what we do with this. My first big NIH grant was go get every human disease studied using these microarrays. Just go get them all. Why do I have to focus on any one disease? So I collect experiments like this where there's disease samples and normal samples, right? So if you studied metastatic cancer, not metastatic cancer, that doesn't count. You got to have normal in there too. Now, it's pretty easy to use text mining, human eyeballs to figure out disease. It turns out it's really hard to tell if someone looked at normal samples, okay? There's 200 words for normal, right? Normal, vehicle, wild type, time zero, margins, control. Right? Those are the top 6. We wrote a paper on just the 200 words people use, so people understood that we could do this. Now, of course, I already showed you, though, for any one disease, you don't just have one data set. You have dozens, hundreds, thousands of them, and we start to do meta-analyses to put these together. And what's the first use case that we had in our lab? We realized we want more diagnostic tests in medicine. We want more blood tests. There are a lot of diseases, including breast cancer, cervical cancer, or certainly ovarian cancer, where we don't have a good blood test. We depend on physical exam, inaccurate imaging. We want blood tests for more conditions. And so our naive kind of central dogma thinking was, if we got tissue, and the tissue has RNA up, which we can get to with these microarrays, then maybe the protein is up. You know, RNA goes to protein. And if enough protein is up in that tissue, the diseased tissue, maybe we can detect some of it in the blood, kind of serum or plasma. Really naive thinking here, right? Seems to work. So then we start to turn our crank. Oh, we can get candidates. We're going to get more and more candidates for blood tests. And we hit upon one for acute myeloid leukemia, AML. Not a good leukemia to have. They talk about putting a bone marrow into you for AML. And we figured, oh, we got a blood test. We have all our data. What are we going to do next? Well, let's go test our computational predictions in the serum or plasma. So I love Stanford. We go to our Stanford Cancer Center, and we said, do you have any saved serum or plasma on patients with AML? And they said, you know what? I'm sorry. We saved all the leukemia cells, all the cancer cells. We really didn't save any of the liquid around the cells, the serum or the plasma. It turns out a lot of freezers you need for that. We're at Stanford. We said, okay, well, they said, you're an academic institution. Go get IRB approval. Go put up the posters. And in about a year, you should have enough patients to test your computer prediction. What do? We just Google for it. And we find samples this way now. Conversant Bio, browse by disease. I see pictures of Vacutainers. I'm in the right store, right? Okay, what do you want to buy today? Bladder cancer, brain cancer, breast cancer? Oh, here's leukemia. And what exactly do you want to buy? Peripheral blood? You want to buy bone marrow samples? Okay, well, I see plasma here. Click on that one. $55 each. How many samples do you want per patient? What disease subtype? Add to shopping cart. This is for real. This is here. This is now. I'm not making this up. This is not the hard part. Where she's asking, do these samples come from? What is this, right? Turns out big hospitals in the middle of the country, even in big cities, the minute that a blood sample is ordered by a doctor or the nurse, they run some blood tests, but it sits in the refrigerator for 48 hours, right? Waiting for the doctor to add more tests. But at the end of 48 hours, they gotta make room in the refrigerator. They gotta throw the sample out, and something magically legal happens on its way to the trash. That becomes a discarded human sample. You strip off the sticker, put on new stickers, don't put any patient identifiers. I, as a customer, have to agree not to identify these people. I truly don't care who these people are. And this shows up on 72 hours on dry ice. This is not the hard part anymore, right? There are hospitals, there's companies that come in and organize all this, waiting for you, just waiting for you. So we turn our crank and we said, let's do this. We got one for medulloblastoma. We had a pathology test. We had some for pancreatic cancer. And then we hit upon this disease, preeclampsia. I hate this disease. My wife had this. My daughter was born super early. She's totally fine now. And this weird disease, preeclampsia, probably none of you have heard of this disease unless you watch Downton Abbey. One of the characters dies. Spoiler alert, sorry about that. But that's the only reason why we know this disease in America anymore, is because one of the characters dies of eclampsia. And preeclampsia is high blood pressure during pregnancy. It's still how husbands lose wives around the world, and there's no blood test for this thing, right? I thought that was really annoying. Leads to a lot of babies being born early. It's nothing we can really do about that. So how would we do this? Well, you just go to the databases. Here in Array Express, you can just type in preterm or preeclampsia or premature. You can see how many, 200 pages of just data sets waiting. You get to pick and choose. Do you want the muscle? Do you want mice? Do you want humans? Pick and choose the data sets you want. Do a meta-analysis. Figure out what's in common. You can, Venn diagrams should be spinning in your head. I don't need to show you those, right? That's the kind of thing we do. And we hit on markers. Here's one called hemopexin, and it's upregulated, normal versus preeclampsia. By the way, you can buy all those samples off the internet because hospitals in Cambridge, Massachusetts just sell you pregnancy samples for $45 each. Now, I'm not saying that's the final answer, but that gives you enough data to convince your own doctors at your own institution to trust you with their samples, and you add more value. Just keep de-risking it as you go. And at the top now, you see Linda Liu is a grad student started. Bruce Ling is a faculty member, Matt Cooper, and a company logo. And I'm going to show you this. You can see a couple of these arrows. And this, to me, is the new way to be an academic. I'm showing you at the bottom here that this company then got spun out and got $2 million of seed financing to keep it going. At the top is the arrow that I keep believing in. You start with the unmet medical need. Public data available. Design the algorithms. You get internal seed grant funding, $50,000 Spark grant just to get ideas off the ground. Spin it out. Technology seems to work. Seed investors go in. I'm not showing you $2 million to brag about it. I'm showing you $2 million because I firmly believe this is the new way science is going to get funded in academia in the future. You get it as far as you can in your lab, and the science continues in a startup company. They're doing science. It may fail, it may succeed, but now external private dollars are taking this forward. And they may live or die, but we're going to do a hell of a lot more of these now. Do you know how hard? You know how hard it is to get a new $2 million NIH grant. right? Sometimes when the story is de-risked enough, it's easier to get the money from Sand Hill Road than Bethesda, okay? And that's what I'm talking about now in this story. And this is now where you can see students get involved. And I know all about postdocalypse, right? It's about creating your own future here, right, as a postdoc or a grad student, right? That's the point here. So that's diagnostics. Let's talk about therapeutics for a second. As we're collecting all this data on diseases, it turns out major labs like the Broad Institute and others are studying the drugs. And what they do is they take cells with drug, without drug, with drug, without drug. This was a paper called the Connectivity Map. It was in Science back in 2006, 2005, and then they expanded that into a project called LINKS. And literally taking cancer cells or ES cells or many other types of cells with drug, without drug. So on the left, we have with disease, without disease. And on the right, we got with drug, without drug. They're not even the same cells. And what we did is we started to figure out how do we put these two together. And the reporters called this Match.com for drugs, okay? It's their idea, right? So, you know, Match.com, you find, you know, a spouse, or you find a date. And what's the famous saying? Opposites attract. So this is, again, naive informatics here. If I've got a disease on the left where this gene goes up and this gene goes down, and I can find a drug on the other side that can make this gene go down and this gene go up, maybe there's a match. Instead of 2 genes, imagine 20,000 genes. I'm not going to show you any formulas. You can imagine how you can do Pearson correlations and all the rest there, right? So we start to turn our crank, and we start to get ideas, and we talk to collaborators, and we can figure out, well, which diseases can we target on campus? But now, I've learned, if I really, really need a drug tested, well, I would love to run a clinical trial. But before you run a clinical trial, you got to have the animal data, right? Mouse data. And when I really, really need to get a mouse study done today, some of you are laughing, you know what I'm going to show you. We go to assaydepot.com, right? Kind of sounds like Home Depot, and you're right? Which aisle do you want to go into? Biology, chemistry. That DMPK is drug metabolism pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology. And toxicology. Okay, let's say we want pharmacology. What type of mouse do you want to run today? Bone, cardiovascular, dermatology, diabetes, genitourinary, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. What mouse do you want to run? Well, I'm a really part-time diabetes guy. Here we go. Let's click down. Here, I can order an OB diabetes mouse model, 16 mice. Maybe we want 2 groups of 8 or 4 groups of 4. If you read this paragraph, you can see this mouse has been eating a lot and getting diabetes since 1951. This is the standard mouse model for diabetes. Kind of teaches you there. And in 28 days, they'll give you all the fasting blood sugar, the insulin tolerance test, the glucose tolerance test, whatever you need in 28 days, you got all the data back. And you can send whatever drug you want to test blinded to them. Now, what am I covering up here? The price. $9,000 for this service, 9-week turnaround time, and literally add to shopping cart. This is for real. I can order any mouse model I want off the internet, right? And I'm showing you Assay Depot, Science Exchange, you just saw from Barbara. You can order these off the internet, but you got to trust these companies, right? You got to trust these companies. Yeah, Assay Depot has a challenge. Science Exchange is doing a reproducibility study. So all Assay Depot is a clearinghouse. They put you in touch with what are called contract research organizations, so they don't run it. But what's happened, CROs have been around for decades, but what's happened is because of websites like this, there's price transparency, the price comes down, even we can afford this in academia now, right? Now, actually, in reality, some of you with sharp eyes, you'll say, well, this is run in China. Actually, when you hit add to shopping cart, that may be a problem for some of you, and that's not a problem for me. But when you actually hit add to shopping cart, you're presented with 100 companies that are willing to do this experiment for you. Do you want FDA approved? Do you want ISO 9001? It's just like Amazon. You click, and you get, I'm a customer, right? I'm a fan of these guys, but this is a new way to do the science. You got to care about the quality. Now, some of you are wondering, boy, you're about to put this into a human. This mouse data better be really reliable to trust. I'll simply answer, if I really need to know whether this drug is working in the mouse model, simply order 2 of them from 2 companies at this price. Hell, at $9,000, order 10 of them from 10 companies, right? How many mouse researchers really, when they get their experiment to work, they go across the hall to get a complete independent team to reproduce their answers? You know nobody does that in academia. So what I suddenly can do now is convert the quality problem as a function of dollars. You have more dollars, you get better quality, more independent teams reproducing your research as much as you want now, right? So I can make the quality problem completely disappear. I got 2,000 breast cancer labs giving me data, and I have as many labs validating as I want with a credit card today. Of course, I'm most famous for my the one that came out in Science Translational Medicine. Our anti-inflammatory topiramate, a seizure drug, had anti-inflammatory properties for treating inflammatory bowel disease. These are my famous rat colonoscopies. Not enough of you are laughing here, so let me explain the geometry involved. This is a colonoscope, and this is a rat. And this is what you'd love to outsource, right? Why learn how to do this, right? These are my famous rat colonoscopies. 40 megs of video on the Science Translational Medicine website. Mouse, normal rat colon, rat with inflammatory bowel disease. Mouse all better, rat all better with topiramate. We didn't know how to do the experiment. We learned along with the contract research organization, right? We have to learn everything. Does it dissolve in DMSO? Do you do this? Do you do that? We have to learn everything about the protocol except touch the rats, right? We had to design everything else. Now I'll show you another arrow, right? We start with unmet medical need. We always, in my lab, start with unmet medical need. We never start with algorithms or methods. We never get anywhere that way. You got to see what's needed in the field. Public data available. NIH funding, my grant got us the data. Analyze it. We had a company spun out of Stanford. We got an ARA grant stimulus, and then $3.5 million from a private venture capitalist. Again, I'm not showing you $3.5 million to brag about it. I'm showing you this is the new way to do science at academia. Get it to a point, get the science level papers you want, keep it going. File the disclosures, file the patents, not to be greedy about it, but if nobody's going to pursue that patent, it's up to you to take it forward now. And that's where I believe our responsibility is. We got some new data coming out. I'm not even going to go into too much details. We got a brand new drug for psoriasis. I don't know if any of you have psoriasis. Everyone knows, everyone treats psoriasis now with these super expensive monoclonal antibodies that wipe out your immune system. Using the same Turner crank approach, we got this awesome drug. It's an old cardiovascular drug. And it turns out this drug actually fixes the skin cell. The immune system is still there, but the skin cells are all better from nothing down to, this is going to be the new dose, full dose, and the immune system isn't even touched. This is going to be one of the first small molecules that fixes psoriasis without actually being an immunosuppressant. And again, I don't know much about mice. This is done in collaboration with Mazen Nasrallah, who's in my lab, Peter Marinkovich, Martin Wingate. Yeah, this mouse doesn't look so happy. This one looks a little bit better, I guess. The tail looks a little bit better. But my favorite, well, we've done like a dozen of these. This is my favorite one, came out in the paper a couple of days ago. I'm just going to end with this kind of thinking here. This is a mouse where if you knock out P53, RB, or P130, it develops small cell lung cancer. Now, humans have to smoke to get small cell lung cancer. This is not a good cancer to have. 5% survival rate in 5 years, okay? And this is small cell lung cancer. On the left, you can see these kind of nodules. So humans smoke, this mouse gets its own lung cancer. It's not a xenograft model, it's getting its own lung cancer when you knock out these genes. And the mouse on the right is the same kind of mouse treated with an antidepressant, imipramine, okay? It's a tricyclic antidepressant. It's still in the pharmacy. We don't use it anymore because we have these serotonin reuptake inhibitors that are a lot nicer to use. This one has some kind of annoying side effects. This one makes you sleepy, and in some people can widen your QT interval and might set you up for an arrhythmia. Actually, neither of those two side effects sound as bad as having small cell lung cancer. 5% survival in 5 years. And here, the cancer is melted away. It's gone. It's gone. Now, we love these repositioning stories, finding new uses for drugs, but this was a doozy for us. We went from computational prediction. It's working cell lines in a dish. I'm not even going to show you that. It's working in the mouse model, and 15 months after the computer prediction, we get IRB approval. We launch a clinical trial at Stanford. And this is the way to do it. I refuse to believe it's going to have to cost $1 billion in 10 years for every one of these drugs. If it does, we're in for a disaster. It's going to come down to all of us as academics to figure out these new drugs, whether it's new drugs or new uses for old drugs. I think this is going to be one way to do it. And you know what? This whole idea of finding new uses for drugs, instead of finding them by accident, how about we start to find some of these new uses on purpose like this? And I want to show you this. Why am I in this business? It's emails like this once a week, right? So this is an email I got October 20, 2013. Hello, my name is, I need help. I'm an Italian man. I live in Sweden. I'm 48 years old. I have these kids, all the same wife. And on June 5th, they told me I have small cell lung cancer. I have all sorts of different metastases. I know I will not live longer than a few months or 1 year. I get an email like this every week from someone around the world who wants to just be part of this trial. Because there are a lot of cancers out there, there are a lot of diseases out there, and they all need attention. And if you believe the pharmaceutical industry can do it all, they cannot do it all. It's going to come down to us. And I'm telling you, the data is sitting there on the internet, waiting for you, waiting for you. I don't even know if this guy is still alive anymore. right? This was back in 2013, and this is what's driving us. Now, if you ask me, what's my next big bet? I thought Barbara would talk about this, but she didn't really. Half of my lab is spent on, to me, the next big open data, and that's clinical trials data. Steve Goodman in IOM, I was at both of his IOM meetings. What's a clinical trial? The most frickin' expensive experiment in the world. Billions of dollars, half of them fail, and we don't even write papers about them when they fail, right? God forbid we even release the data. It's gonna change. It's gonna change. Europeans, EMEA is requiring raw clinical trials data to be released. FDA may get there someday if the IOM report really works out. And I'm imagining all sorts of new directions for clinical trials data. Maybe we can figure out, why do you think this drug really failed? It seems to be working in a quarter of these people. And I can tell the quarter of these people with this Goomba marker. And it's the same Goomba marker in this clinical trials. Why do I even have to run a new study? I can test and I can learn and test in the same in silico marker set, right? I mean, I can find new patient subsets. I can do meta trials. I'm imagining what I can do with this. Again, I'm trying to skate to where the hockey puck is going. I see it going this way. I'm going to build all sorts of methods to figure out clinical trials data. I happen to run this one for NIAID, one of the NIH institutes. We give out 100 raw clinical trials right now. You can go download, play with clinical trials. Each one's 200 tables. You learn what a trial is all about. Go sign up for an account. And the data just keeps coming, whether it's wearables, whether side effect data, our electronic health record data, EPIC, Cerner, pharmacogenomics, cancer genome atlas, all the rest. So really, for take-home points, what I would say is I have to convince you, I hope you believe, there's plenty of high-quality data already available. Some of it is public, some of it's private. We have electronic health record data, we have things that we don't give out, but look how much is actually public. Don't wait for perfection. I know it is bad, it is always getting better. You will always argue, though, it's better this year than it was 10 years ago, right? It'll probably be better 10 years hence from now. But we'll still have some way to criticize it 10 years from now. Don't just get in the battle of criticizing. Think about what you can do with the data today in its current form. Use and intersect data to ask new questions. You don't step on toes that way, it turns out. And it turns out the intersecting questions tend to be higher impact than just the itty-bitty little data sets. And academia and industry are compatible. The science can and will continue in industry. You're not going to be able to do everything in your lab. To close it out, I'm leaving Stanford after 10 years, moving to UCSF. I'm heading this new Institute for Computational Health Sciences. We're going to have to slum it out in this brand new building in Mission Bay until they build our brand new building in Mission Bay, right? Ironically, right in between where Illumina is and the new hospital in Mission Bay. I'm going to be recruiting a dozen new faculty over the next 5 to 6 to 7 years. And why would I move to UCSF from Stanford? Yeah, there's a building. Yeah, I get to recruit faculty. But they put me in charge of something called UC Health Informatics. Basically, University of California, San Francisco is one of 5 University of California medical schools. They all have electronic health records. And I get to oversee the build-out of a common electronic data warehouse, secure, de-identified, of 13 million patients seen at any UC hospital. It's about 4% of the US population. Imagine what we can learn from just patient data, teaching institutions to improve. My experiment of the future is to learn from 3 and teach the other 2 and just keep rotating, right? Improve an entire health system. That's why I'm moving to UCSF. Can't do any of this work with a long list of collaborators. Ones in red here are the ones that really helped the most. The folks at Northrop Grumman who helped me with import, Alana Sweet-Cordero and Julian Saj and their team on the mouse cancer work, Jay Pashvik on the gastroenterology work. Can't do any of this. A long list of collaborations and support. Endowment support from Lucio Packer Foundation for Children's Health. My lab is blessed with 17 NIH grants from these 9 institutes of NIH. These 4 give me more, these 5 give me less. I still love them. Marshall Dimes, HP, HHMI, a lot of cancer-specific foundations. I always thank my admin and tech staff, would never get a grant or paper out the door without them. And I always thank my wife, molecular biologist, biochemist, who reads every major paper or grant that comes out of my lab, who starts companies with me now, and lets me go all over the place to give talks like this. Thank you very much. I think I went over for time. Maybe they'll tolerate a question.
Any questions?
Burning. Over here.
Yeah.
So most of human diseases are complex interplay of multiple genes. So biology is much more complex.
That's right.
And how this relatively simple computational prediction from microarrays, I assume it may some low-hanging fruits it's come out,
Absolutely right.
But for most of them it looks unlikely.
That's right. So it's really tempting to think about it from the engineering perspective. And the engineering perspective is we have to completely understand this disease before we can come up with something or recommend something. And meanwhile, patients come every day with these diseases. So actually in medicine, it's about comparing to what we can do now. And what we can do now is pretty terrible for many diseases. So you could call it low-hanging fruit, you could call it a marginal improvement, but that is a world of a difference for some patients. We don't have to wait for perfection, perfect knowledge for these diseases, perfect data. You're right, it's completely imperfect. You're absolutely right. But for $9,000 to test it in a mouse, I'm going to do that before just making it perfect, see if it works there, and move it on.
Okay,
Thank you very
again.
much.